Good morning or good afternoon, depending on where you are, and welcome to today's webinar. My name is James Esley McDonald, and I'm part of the World ADC team, and we're delighted to have two speakers from Lonza on our webinar today, sharing their thoughts and work on engineering and manufacture of second-generation ADCs, tools and techniques. Ramon and Christoph will be speaking for around about 40 to 45 minutes, and then we'll have the remaining time available in today's session for audience question and answer. So you all know there will be a recording available after the session, and a copy of the slides can be requested from one of the Lonza team, but we'll provide that email at the end of the session. If you have any questions as we go, please do submit them in the chat box, and we'll try to get through as many as possible at the end of the session. But for now, and without further ado, I would like to pass over to our first speaker of the webinar, Ramon.
Thank you very much, James, and good afternoon and good morning, I guess, everybody. Thanks a lot for attending this Lonza webinar. My name is Ramon Gomez, and I am a principal scientist in Lonza's cell engineering group here in Cambridge, in UK. And I will be talking during the first half of our presentation about the site-specific conjugation vectors that we have created in Lonza for the generation of ADCs. So my first slide is the compulsory disclaimer that you will have time to read later on your own time. And although I'm sure that most people in this webinar know what antibody-drug conjugates are, just in case, I have also put an introduction slide for those of you that are not familiar with the concept of ADCs. So antibody-drug conjugates are molecules that combine the specificity of monoclonal antibodies with the potency of cytotoxic drugs. If you consider the therapeutic window of a drug as a toxic dose over the efficacious dose of that drug, in chemotherapy, the therapeutic window of the anti-cancer drugs that are injected in patients is very narrow because cytotoxic drugs trigger systemic toxicity at very low concentration. However, if you attach that drug to an antibody through a linker, the antibody will transport the drug to the cancer cell, where the antibody will bind selectively to the overexpressed antigen in that cancer cell, and the antibody bound to the cytotoxic compound will be internalized, and inside the cell the toxin will be released and will kill the cell. So there is no systemic toxicity. The drug is only released where it is needed, and doses can be higher, the therapeutic window widens. Not only that, but the antibodies can also be loaded with potent cytotoxic compounds, which have much higher potency than the known anti-cancer drugs, because we know that they will be taken to where they are needed and nowhere else. At the moment, there are 4 ADCs in the market: Kadcyla, Cetris, Mylotarg, and Besponsa, and there are more than 300 antibody-drug conjugates currently in clinical development. At Lonza, we have an end-to-end offering when it comes to ADCs, and we achieve that by combining our facilities within our network. So we manufacture the antibodies in Slough, for instance, in UK, and we synthesize the payload in Visp in Switzerland. We combine them together, and then we do the bioconjugation in Visp as well in Switzerland. This way, using Lonza network, we simplify the supply chain and we save resources in activities like coordinating and tracking all these processes. We also have a drug product facility in Basel in Switzerland, where things like formulation, packaging, etc. are assessed. Our commercial bioconjugate facility in Visp is currently supporting 2 commercial ADCs and 6 late-stage ADCs, and has capacity for new projects. Every year, more than 50 batches are released with nearly 100% batch success rate. And obviously, all this is inspected by the relevant bodies like the FDA, Swissmedic, etc. So currently, all the ADCs that are in the market are conjugated to a drug in a not specific manner. They use either the amino group of a lysine or the sulfhydryl group of a cysteine. If we consider a typical IgG1, this antibody, this molecule, has 4 interchain disulfide bonds. So when the molecule is reduced, there are 8 sulfhydryl groups exposed to which linear linker drug can be conjugated using different chemistries. And that renders a very heterogeneous product with ADC populations that can vary from antibodies that have no drug conjugated to them to antibodies that have 8 drugs conjugated to them and everything in between. For lysine, is even more pronounced because on average in an antibody, there could be more than 90 lysines. So the heterogeneity is massive. Even if there are lysines in regions that are more prone to interact with linker drugs, the heterogeneity is very, very high. Normally, higher number of drugs equals higher potency, but it also means higher hydrophobicity. And these, in turn, will lead to potential immunogenic responses, to shedding of the drug, to maybe steric impediments to bind the target. And very importantly, if this ADC has to be manufactured, there will be significant challenges for the consistency of the process and for the characterization of the product. And it also will be difficult to understand the relations between the site and extent of the drug loading and the ADC attributes like efficacy, safety, etc. So it follows that site-specific conjugation to an antibody will create a much more homogeneous product without any of the disadvantages that I have mentioned. And this way, a higher therapeutic index is achieved. So I think it is safe to say that site-specific conjugation is going to be a must in the ADC field. And with that in mind, in Lonza, we have created new site-specific conjugation vectors for our GS Xceed system. And what these vectors will allow is an easy subcloning of the variable regions of a heavy or a light chain in a single ligation step into the vector. And this way, a very fast construction of conjugation-enabled antibodies, either chimeric or humanized. Obviously, the DNA sequences are gene optimized. So to create our site-specific conjugation anchoring points, what we did was introducing cysteine mutations on the constant domains of the antibody. And we chose cysteines because they usually are not found reduced on the surface of antibodies. And they can be modified as well using a wide range of different chemistries. Cysteines are normally joined together between their side chains via disulfide bonds. These bonds must be reduced to sulfhydryl groups to make them available for cross-linking by the reactive groups. I have added here some examples like maleimide, which is very popular, and it reacts at near neutral conditions with sulfhydryls. There are also haloacetyl cross-linkers that contain iodoacetyl or bromoacetyl groups. Pyridyl disulfide react as well with sulfhydryl groups, etc., etc. For our proof of concept, we chose trastuzumab as the model antibody. As probably most of you know, trastuzumab is an antibody that targets epidermal growth factor receptor 2, HER2, which is a gene that is amplified in around 30% of early stage breast cancers. And trastuzumab is a very well-known and characterized molecule. So we thought it will be a very good model antibody for our experiments. And we introduced the cysteine mutations in a rational way following different criteria, like choosing residues with similar psychochemical properties, or B, residues with a small non-hydrophobic, non-charged residues like serine, valine, threonine, alanine. Obviously, we wanted them in the constant regions of either the light chain or the heavy chain. They should not interfere with the target binding domain to minimize the risk of disrupting biological activity of the molecule. We didn't want them to create intra-chain hydrogen bonds that will lead to the alteration of the local environment and this way of the properties of the protein, of the antibody. The solvent accessibility surface was important for us because we wanted those residues exposed enough so they could react with a linker toxin, but not too exposed, because then they will be very prone to aggregation. And in the other hand, we didn't want them very hidden, because then they wouldn't be able to react with the molecules we wanted. So when we took all those criteria in mind, that left us with a limited number of residues. We mutated them to cysteines and then tested in the wet lab all the variants, looking at the yield of the antibody, the aggregation, fragmentation, how it bound to its target compared to the control, to the trastuzumab control, the drug-to-antibody ratio, stability, etc. So the next step was to conjugate our vectors, site-specific conjugation vectors, to a toxin, and we chose for our proof of concept monomethyl auristatin E using a valine citrulline linker and a maleimide bridge. For when we used this molecule, this drug, and as I have said before, and we did the non-specific cysteine conjugation, we obtain, as you can see in this profile, a very heterogeneous set of products. This is the reverse phase chromatography profile. So we have drugs that have no drug whatsoever, antibodies that have no drugs, or antibody that have 8 drugs, and everything in between. However, when we use our site-specific vectors, as you can see in this molecule that has 2 drug molecules attached to the heavy chain, we have a profile like this. The light chain doesn't move because it doesn't have any drug attached to it, but the heavy chain moves to H1, so it has one payload attached to it. Obviously, the antibody is reduced here. And the populations in between are all almost negligible. So we are achieving a DAR of 2 or very, very close to 2. In this example, with an antibody that has 2 drugs in the heavy chain and 2 drugs in the light chains, as you can see here, the light chain now migrates to L1, so it has 1 payload, and the heavy chain remains with 1 payload. And again, negligible populations otherwise. So we are achieving a DAR of 4 or very close to 4. We were happy with those results, and then we wanted to test the potency of our ADC products in vitro. So for that, we tested them in 5 cell lines, 3 that were HER2 positive and 2 that were HER2 negative cell lines. What I am showing here are the results of the SKBR3 cells that are a type of breast cancer cell overexpressing HER2, which is the target of trastuzumab, as I have said. And our ADC molecules were conjugated to MMA, as I have said, and the experiments were done in triplicate in a concentration range of around 33 nanomolar to 0.5 picomolar. And the control was trastuzumab not specifically conjugated to MMA. And this will be the control in all the slides that I'm going to show you. So as you can see here in the SKBR3, our variant that has 4 drug molecules conjugated to it gave an IC50 well into the sub-nanomolar range. So it was very good. Our other 2 variants here, these examples, with 2 toxins conjugated to them, either in the light chain or in the heavy chain, had a very similar profile to the positive control, with IC50s also well into the sub-nanomolar range. So this was very good. We also try our HER2 positive cell lines, as I have said. We try SKOV3 and BT474, and the profiles were very, very similar to this one. So there was a very good response. And then, obviously, we had to try negative cell lines. And what I'm showing here is MCF7, which is a HER2 negative cell line. And as you can see, there is no effect. All the cells survive. So by doing this, what we are proving is that our variants, our vectors, are acting through a specific receptor-mediated internalization mechanism, which is very good. They are very, very, they are not, they have very precise, they localize where we want them to localize, basically, to the HER2 and nowhere else. We try this as well with another negative, HER2 negative cell line, MDA-MB231, and the results were the same. There was no response whatsoever, so all the cells survived. Then the next step was to try the same experiment, but in vivo, to see the efficacy of our ADCs. And to do that, what we did was inoculate subcutaneously SKOV3 cells that are human ovarian cancer cells expressing HER2 into a thymic nude mice. So we just basically put a xenograph in them with a tumor. And then when the tumor reached an average volume of around 200 cubic millimeter, we injected 3 different single doses, 1.5, 3, and 6 mg per kilo of the ADCs. And as you can see here, at 6 mg per kilo, again, our variant that has 4 drugs triggers a remission of the tumor and certainly a pronounced slowdown of the tumor. It is even better than the positive control. And then our other 2 variants, as an example here, with just 2 drugs in the light chain or in the heavy chain, they haven't got such a pronounced effect, but they certainly slow down the tumor, which is very, very good. We saw very similar results in the 3 mg per kilo doses and a mild effect in the 1.5 mg per kilo doses. The next thing we wanted to try was the stability of those conjugates. So to do that, we incubated them in human serum at 37 degrees, and we took samples of that serum at different time points between 0 and 72 hours. The ADC was purified using HER2-bound Cepharose beads. It was washed and eluted from the beads, and then the DAR, the drug-to-antibody ratio, was analyzed by mass spectrometry. And as you can see here, for our variant with 2 drugs in the heavy chain, or our variant with 4 drugs, after 72 hours, they have shed only around 20% of the drug, whereas for the positive control, we have lost after 72 hours almost 50% of the drug. So again, they proved to be superior, better than the positive control. All the data that I have shown so far has been using monomethyl auristatin E. So that's what you can see in the top row. This is what I have shown you. But obviously, we wanted to investigate an alternative payload. And during the time we were doing this study, we were collaborating with a customer that wanted to try a new toxin. So although we were thinking about using just trastuzumab control, we decided to use our variants as well and 2 different linkers. So we wanted to use the valine citrulline that we had used with a new toxin, and then Lonza's branched linker, LL2. And as you can see here, when the new toxin is conjugated to valine citrulline, the unspecific conjugated trastuzumab, so this is the control, gives almost no response whatsoever, whereas the variants with a DAR2 and with a DAR4 have a mild response. Interestingly, the DAR2 seems to have a more pronounced response than DAR4. And then when we use the branched Lonza linker with a new toxin, as you can see here, the control gives a better response, and the DAR2 and the DAR4 variants give a similar response. Again, I would say the DAR2 gives a better response than the DAR4. I guess that what I want to show in this slide is how important it is to choose the right toxin and the right linker, and what a difference this makes. So we have created site-specific conjugation variants that have no effect in aggregation. Pre-conjugation, the variants show monomer purity around 97%, and post-conjugation, the monomer purity range between 95 and 90% and 98%. They have no effect in expression. All variants had comparable concentrations to trastuzumab in 10-liter bioreactors. No effect in melting temperature, which tells you how stable the antibody is pre and post conjugation. And as you have seen in the in vitro and the in vivo study slides, the variants showed good specificity and good internalization. So what we have at the moment in our GS system family of vectors, we can create monoclonal antibody constant framework libraries with our picon vectors, either by fusing a variable heavy or a variable light domain. But by adding these new tools to our GS system, what we can do as well is combine those picon heavy or picon light with the site-specific conjugation vectors. And this way, what we will do is create a conjugation-enabled library for 2 payloads in this case. But also, if we only conjugate the site-specific conjugation light and heavy, we will be creating a conjugation-enabled library for 4 payloads. And I guess I didn't want to leave without saying that these vectors, although we have been talking about ADCs all the time, they can be used to create another immunoconjugates. So we can use them to conjugate a radionuclide. We can create antibody RNA conjugates, antibody antibiotic, protein antibody conjugates. We can conjugate fluorophores, etc. And these are all the slides I have. And I'm going to hand it over now to my colleague Christoph in Switzerland. Thank you very much.
Thank you very much, Ramon. My name is Christoph Klochtz. I'm representing the bioconjugates group located in Lonza FISP site in the beautiful Swiss Alps. And I'm going to take you through the second part, the conjugation part of the slide deck. On the first slide of, you see on the left-hand side, a generic engineered cysteine SSC process. And we will go through the parts of this process in the next couple of slides. Everything starts with the process idea, which is initially tested on a milligram scale thermalization pattern. And already at this early stage, it is very important to use fully purified and representative map, and if possible also payload, in order to generate representative results and avoid misinterpretation. From the initial information available and the result of the familiarization can be constructed a production concept. And the first important thing is to add in-process controls for analytics to get control over the process and to generate process knowledge. As mentioned, raw material qualities is already very important at this stage, and I'll talk about filtration a bit later. Then a couple of words to safety. It's pretty clear that ADC work has to do with cytotoxics and highly potent compounds. And certain rules and procedures have to be followed, especially when it comes to cleaning and, for example, to spillage control. Other risks are not that obvious. It's certainly worth checking the resistance of your isolator and laboratory gloves to the destined cleaning solvents and co-solvents. And any non-compatible items, such as seals or O-rings or tubing, can in fact lead to spillages or leakages. Another risk added by the solvents is the risk of fire. And as you can see here on the right-hand side, that is a flashpoint diagram of isopropanol water mixtures that tells you that concentrations of larger than 30% isopropanol produce flashpoints of 25 degrees ambient temperature or lower. So there is actually a fire hazard. And measures that can be taken are either running your isolator under nitrogen and or using ATEX certified equipment in the areas where this may happen. Now to the process itself. The first step, the first real chemical transformation after pooling a map, diluting it and pH adjusting it, will be the reduction. And the typical reagent is very prominently shown here. It's TSAP-HCl, and alternative reagents would be DTT, dithiosrytol, for example. And a generous excess of this reagent will lead to the deprotection of the protecting groups on the engineered cysteines and simultaneously reduce interchain disulfide bridges. Some types of aggregate dimer and trimers can also be cleaved, as can be trisulfide bridges if present in the map. And as a phosphine reagent, the buffer components should be checked for their compatibility. The driving parameters are listed here for this reduction reaction, and it might be worth checking the free thiol content, both in the non-reduced antibody and after full reduction, in order to check on the capping status and the reduction status of the map. Next step is the cross-flow filtration, and there are typically two cross-flow filtrations in a site-selective conjugation process, one after the reduction step and one after the conjugation step. The one after conjugation will take away the free drug. This is shown here on the bottom right. That is a free drug depletion chart. And the membrane materials which are typically used in the TFF membranes will be polyether sulfone, PES, and regenerated cellulose. And regenerated cellulose can offer advantages with respect to the solvents, co-solvents used in the conjugation reaction. There are a couple of molecular weight cutoffs, 10 kilodalton, 30 and 50 kilodalton available, different screen types, different suppliers, and there is quite a lot of optimization potential here. Peristaltic pumps will usually generate worst case retentates, while membrane pumps on larger scale are normally a bit milder on the maps and products. Standard settings are shown below here, and if you apply those, you can optimize, for example, as shown on top right, by modifying or varying the TMP, transmembrane pressures at different feed flows. You can optimize the filter rate flux, which gives you then short and efficient processes if you have a very good and optimized filter rate flux here. A suitable protein concentration for experiments is usually chosen at the beginning based on the available map and drug linker and the available smallest size of TFF membrane or the vessel size, and can be optimized later. Optimization potential is present for the protein concentration and the membrane loading, and this for the reason as membrane area is really expensive. That will also bring cleaning and reuse of membranes into question very soon. Monitoring the aggregation behavior is also very interesting, both on the non-reduced stage, after the TSAP reduction, and after and during the TFF, because this can tell you something about the stability of your protein. After the TFF, the next step is the reoxidation, meaning the reconstruction of the native disulfide bridges and not touching the engineered cysteines. Typical reagent here is DHAA, dihydroascorbic acid, here shown as a monomeric structure. In fact, it is, when solid, a dimeric structure, shown on the right-hand side, which has a very interesting dissolution behavior in water. It's a fast-acting reagent, producing ascorbic acid during the reoxidation, and the ascorbic acid can slightly impact on the A280 measurement after a reoxidation. It avoids biphasic air oxidation and also the use of, for example, copper sulfate, which is not desired. And a very important feature of this reagent is its pH-dependent and irreversible degradation. You can see that here on the chart on the bottom right. And for example, a solution after 2 hours at a pH of 7 already loses 60% of the desired reactivity. At a pH of 8, already 80%. And moreover, there is literature available that states that under certain circumstances, the degradation products can react with proteins. As development options, surrogate conjugations can be used to check on the target DAR in order to demonstrate good reoxidation. Now, after the reoxidation, the conjugation is actually the next step. And that is the step where a hydrophobic, or usually very hydrophobic drug payload that can precipitate from water needs to react with a hydrophilic antibody. And the antibody can, in this case, also precipitate or form aggregates when confronted with co-solvents. Typical co-solvents involve alcohols, glycols also, and dimethyl acetamide, dimethyl sulfoxide, and the reaction conditions such as the pH, the temperature, or time are usually adapted to the linker chemistry as much as possible. Always keeping in mind that the protein is limiting, for example, if reactions are run near the PI. Using aqueous solutions together with organic co-solvents can lead to mixing exotherms. And density, talking for example of ethanol, density is 0.79 kilogram per liter, DMSO 1.1. That can cause issues on mixing, especially on scale up. So mixing is something which should be looked at. Pre-adding some of the solvent prior to adding the drug linker solution is, for example, an optimization option. The conjugation is terminated usually by a quench, either by setting the pH to a low level or by adding N-acetylcysteine in order to react, for example, with malamides. And this is useful in order to stop any non-selective lysine conjugations, for example. An important task after conjugation is the removal of free drug and cleaning up of the product. And that is easiestly achieved with TFF, as demonstrated earlier. And if that is not sufficient, chromatography or filtration over charcoal can be options that can be applied. Filtration is very important in the production of ADC. And the most critical filtration is usually the one after conjugation and TFF, because at that stage, a significant quantity of aggregates can be available or can be present, and cloudiness. Typical membrane materials for filters are polyether sulfone or polyvinyl fluoride. And the hydrophilization modification of the surfaces are actually trade secrets of the actual suppliers. There are different types of filters. There are high-capacity pre-filters for really cloudy solutions, standard bioburden reduction filters, the higher-grade sterilizing grade filters, usually with 2 membranes, and validation on removal of bioburden. And end-fill filters are usually optimized for low hold-up and low protein adsorption. Again, the combination of the different suppliers, the different materials, different filter types, and pore size combinations allow a lot of optimization work. One peculiar item is that polysorbates, tween 20, PS20, there are others like PS80, tween 80, can show filter-dependent adsorption as shown here on the right-hand side. And this is one item to be observed or one item to be checked for in the final filtration, which contains already the excipients. For process adaptation, filter sizing should be done and constant pressure should apply for filtrations where a pressure transfer is done between two vessels, while a constant flow approach should be used whenever you have a pump filtration with a constant flow over the filter. Typical R&D filters in the size of, let's say, 2 square centimeters to 20 square centimeters cause an issue because those filters are actually flat and they have a different resistance to the incoming flow of solvents or of solutions than the larger folded filters, which are different. So when doing filter sizing studies at an early stage, and when in doubt, oversize the filter for an early stage, and refine the filter sizing study at a later stage using representative production material with representative folded filters and the flushing wetting regime. Formulation is a trade of its own kind, and formulations are tailored to the MAP starter and the ADC drug product form. And for the ADC, that also considers the effect of drug linkers, the stability and degradation pathway of drug linkers. They contain buffer systems, that means inorganic or organic salts, stabilizers such as sugar or sugar derivatives, and surfactants as the aforementioned twins polysorbates. And the polysorbates are there to relieve some interfacial stress and to counteract hydrophobicity issues. Some peculiarities also here. Polysorbates in MAP formulations can actually be carried through into the ADC through all the TFFs. That needs to be tested. Sugars can increase the endotoxin bioburden load. And if phosphate buffers are in play and deep freezing is intended, precipitation of phosphates can lead to local extreme pHs impacting on the stability of the protein. Tris buffer pH goes up when temperature goes down, and container integrity is important when storing under dry ice due to the carbon dioxide. A typical formulation procedure is shown here on the right-hand side. And after the final TFF, the concentrated retentate, which might be interesting to look for the stability of, is diluted to a target concentration and then formulated with a conditioning buffer containing polysorbate and sugars. And in case there are higher concentrations of protein involved in the TFF, the volume exclusion and Donnen effects can lead to a mismatch between the input buffer concentration and the concentration of salts in the formulated product. The process parameters are quite easily defined. The site-selective conjugation process are rather straightforward, and a systematic approach such as a DOE with a statistical work behind it can be started very early in development. This leads to matrices of results from which the behavior of the system can be deduced. However, scale-down model limitations need to be checked for. For example, stirring is very efficient at small scale, and magnetic stir baths can lead to aggregation in this case. The handling of reagents, for example, pipetting with Eppendorf pipettes of cold and non-aqueous solutions might be inaccurate, and the addition speed is usually very fast. Glass and plastics, if transparent, can lead to some photochemistry impacting on the product. Headspace can come into play when there is oxygen and the oxidation is an issue. And other equipments need to be checked for their adaptability for a scale-down model. Hidden critical parameters might include trace metals from reagents or buffer salts, and chelating agents like EDTA can be used to counteract that. In solvents, for example, the water content of amine in, for example, DMA, or the presence of oxidation products can impact on the drug linker. And PS20, polysorbates in general, are notorious for oxidation under light and air. Finally, sample handling can be important because if samples are not measured immediately but stored for a prolonged time before measurement or even frozen and thawed, this might impact on the result set. QC is a very important player on the road from development to production. And in general, it can be said that most of the data should be generated in the development phase, and production in process control should be minimized in order to allow a smooth and neat run-through of the production campaign. On the right-hand side, you see a collection of typical scale-up related topics which occur quite frequently. Some consider, for example, there is stirring issues, stirring tip speeds causing aggregation, layer formation. Then there are addition rates which are different in production than on smaller scale. And there is heating and cooling that does not scale well at all and should be removed in the PD phase if possible. Other items which can offer surprises are supplier changes of raw materials, consumables which are not available at production scale, QA compliance issues, incompatibility if hydrochloric acid is used at an early stage but is not compatible with steel and steel piping. And many of that can change the impurity profile on scale up. So here at Lonza FIS, we do have the laboratories, we do have the teams to do the scale up and process development work on a daily basis. And we have been doing that for quite a while successfully. And with this slide, I would like to end my talk and thank you for your attention. And the next on the agenda will be the question and answer session. Thank you very much again.
Well, thank you very much, Christoph and Ramon, for your presentations. We now have some time for question and answers. So if people would like to submit any questions they have for the panelists into the question box, we can try to get through as many of those as we can during the remaining 15 or so minutes we have before the end of the session. Just while we compile those, we've got another short poll as well for all of you, just while we're taking a moment to do this, but we'll start the question and answer in just a moment or so. So if we just get the question and answer kicked off, the first question actually we wanted to ask was probably to Ramon, which is, Ramon, how does the cysteine conjugation compare to other site-specific conjugation technologies?
Right. So that could be a very long answer, but I will try to keep it as short as possible. So first of all, let me tell you that currently conjugation via engineered cysteines is the main site-specific conjugation technology that is used in the clinic, and is the one that we are most experienced with. But having said that, there are other methods like non-natural amino acids, using enzymatic conjugation, thiobridges, etc. And they all have their advantages and their disadvantages. For example, to specifically conjugate to non-natural amino acids, a completely orthogonal translation system has to be used in the cells with orthogonal tRNA and tRNA synthase pairs, and that will recognize a stop codon, an amber stop codon, I think, and will place a non-natural amino acid where this codon is recognized. So one advantage of this technology is that the amber stop codon can be introduced anywhere in the antibody. However, creating a cell line able to cope with that is very, very difficult. I think that at the moment, the easiest way to apply this technology is using cell-free expression, which is a technology that is not available to everybody. I have also mentioned enzymatic conjugation, for example, microbial transglutaminase, which is quite precise, but in this case, the antibody has to be deglycosylated, or the asparagine 297 has to be mutated to glutamine, or transglutaminase recognition sequences have to be introduced in the antibody. So the process could be quite laborious. Other enzymatic conjugation is transpeptidases that recognize a specific tag that has to be added to the antibody. And then through a couple of steps, a linker drug can be added to it. Although the problem with this is that transpeptidase-mediated reactions are quite reversible. Thio bridges, I have mentioned, is a proprietary technology, I think, at Zenas, and it uses the interchain disulfide bonds, and by reducing the molecule and re-bridging those exposed to fibrils with other molecule other than maleimide, they can conjugate linker drug. I think that they have a license deal as well with other US biotech company, so it's not accessible to everybody again. And cysteines, cysteine conjugation has several advantages, like the fact that antibodies with cysteines can usually be expressed without problems in mammalian cells, and the conjugation, as Christoph has shown, could be very straightforward. I know that in the past there have been concerns because depending on the position where the cysteine is introduced, it could be prone to exchange, to thioether exchange with other free cysteines that might be in the serum, with glutathione or with albumin. But technology has advanced quite a lot, and there are papers that are showing and experiments that are showing how positively charged environments near that cysteine help to stabilize the linker. I think that it opens the succinimide thioether ring, and that positively charged environment could be introduced through the linker or maybe neighboring residues. There are people also using sulfone linkers or replacing maleimides with acetamides. All these seems to be contributing quite a lot to serum stability, so I don't think it's such a big issue anymore. I think that answers that question.
Yeah, that's perfect. So the next question we have is for Christoph. This is from Daniel. He's interested to know, how long does the Ellman test take?
Hello, this is Christoph. So an Ellman test would probably take about 30 minutes, roughly 30, 40 minutes, probably an hour. But this is something which is usually done in the R&D phase only. And it is quite known that Ellman is not really accurate. And for that, sometimes it's more useful to do conjugation work in order to see what exact DAR you get at this stage from the free thiols you have. But this is nothing you would, for example, do in a production campaign as an IPC. That is a typical thing from which to generate process understanding and to refine the development.
Okay, great, thanks. And then Daniel actually had a follow-up question related to the IPC. Is there an IPC for the TSEP reduction to show the amount of MAB reduced?
TSEP is applied, as mentioned, in a generous excess. So generous means somewhere between, let's say, 20 and 50 molar equivalents, and that's why the TFF is so important. And after the reduction, if you go or try a conjugation with an active reagent at a large excess, you should expect that you have the two cysteines, engineered cysteines, for example, liberated plus the 4 disulfide bonds broken. So you can expect a DAR of up to 10 if you have 4 engineered cysteines to 12. So that would be a way how to quantify on how well your reduction works. Thiol testing could probably lead to the same result, but I would think that it's less accurate.
Okay, and then a follow-up question related to this from Yanis regarding the TSEP removal. After reduction, how is the TSEP inactivated or removed?
That will be done by the TFF. So the TFF, the main task of the TFF after reduction is to remove the TSEP to an extent that it doesn't interfere with the reoxidation which follows on. If there is a considerable amount of TSEP left, this might interfere with your reoxidation, and if the reoxidation is not complete, then you will have overconjugation due to still native disulfide bonds which are not reconstructed.
Okay. And then we have a couple of questions now, both for you again, Christoph, related to the reoxidation. So firstly, from Andrea, why is it that you need to reoxidize the MAB before conjugation?
That is a consequence of what I said. Using a really large TSEP excess reduces both decaps the engineered cysteines and simultaneously removes practically all of the available disulfide bridges. And if you would go then into a conjugation without reoxidation, you would also have the conjugation of the thiols generated from the native disulfide bridges. So at maximum a DAR of 10 or of 12, depending on how many of the engineereds you have.
Okay, great. And the follow-up question was from Terence. With ascorbic acid for reoxidation of the disulfide bridge, what is the propensity for intermolecular dimers?
Intermolecular dimers being formed between two maps, between the free thiols of two maps. This is one thing which can be looked into. This will probably depend also on the concentration on which the reoxidation with dihydroascorbic acid is done. If you apply, for example, size exclusion chromatography to check for dimers, that might be one peak which could show, depending on your exact reaction conditions and, of course, on the map.
Okay, well, thank you very much. Let me move to one of the questions for Ramon to give you a quick break, Christoph. Ramon, the question we have is, what is the maximum DAR you can achieve with the Lonza system? And that's from Alexandra.
Okay, that's an easy one. Is 4. Bear in mind that we have one heavy chain vector and one light chain vector, and in each one of these vectors there is just one cysteine introduced. So being 2 light chains and 2 heavy chains, it would be 4.
Okay, great. And then we have a question for Edward that I was going to probably direct to Christoph. Regarding single-use equipment, could you just comment on your views and perhaps one pro and one con?
Yes, I can. The main pro is definitely that you don't have to clean it, especially when it comes to cytotoxic and very resistant, difficult to clean drug linkers. That is a huge advantage if you don't have to clean it and demonstrate the success of the cleaning. The con is the plastic, which can shed leachables, which is something which you will have to assess as soon as you go near a GMP production.
Okay, thank you. The next question we have is from Lisa. She's asking if you'd be able to explain if the OFAT is giving CCPs.
That is, that's a good question. The critical parameters you should find depending on how exactly you set your high and lows in the for the one factor at a time variations. This will require some initial process knowledge, and this will of course be something which is different from what I said. If you have an idea about what has been in the literature, I think you are quite fine in setting initial, for example, concentrations, initial equivalents, initial temperatures and times. But immediately expecting that you can target critical ranges, that is something. It's a good question. I can't answer this straight away.
That's fine. We can give you Lisa's details to follow up with afterwards as well for a further explanation. The next question we have is that I think you mentioned in your presentation regarding both the antibody and the payload manufacturer and the conjugation actually, they all happen at different Lonza sites. How does this difference in processes all get managed?
This, Christoph, I can comment on this. I've been working on those multi-site projects, and how it works is there will be one designated project manager who coordinates the work between the sites, and he's a contact for the customer. And the main job is to coordinate, for example, the timelines and activities such as, let's say, delivery of antibody from Slough to Visp to have it here for the development work of the conjugation. Then the timeline for drug linker production, which might happen in-house in a different group, or tasks, for example, concerning formulation development at Basel. So everything will be coordinated by one person, the project manager of this intersite or intergroup project.
Okay. Sorry, I was just going to say we probably only have time for a couple more questions, so I'll try and squeeze them in. What's the maximum OEL that you can handle in production?
Maximum. Okay. The risk assessment which is in place for our production facility goes down to a minimum OEL of 1 nanogram per cubic meter, which is pretty low, and that is for the different steps involved. For example, the handling of the payload inside an isolator, the handling of an ADC in the production facility at scale. That is possible. And if there should be the need for having a more potent payload or a more potent conjugate, then this would require an assessment of the risks connected to that.
Okay, and then probably just the last question that we have time for, which is from Vivek. How does Lonza ensure there is no non-covalently bound drug to the antibody?
Non-covalently bound drug to the antibody. So first of all, the cleaning, the purification, as pointed out, either using, for example, the TFF in the easiest case, or column chromatography, or charcoal. This should remove any drug which is just floating around. And there are, of course, very sensitive free drug methods which should be applied by QC to demonstrate and to show that the drug is gone. Then we have the procedure of the quench of the conjugation reaction, which should actually stop non-selective conjugation of active linkers on lysins or other residues. So one part of the development work will be to assure that this conjugation works in a selective way as expected and that there is no such thing. Of course, parts of the work and part of it is, of course, optimizing the different procedures, such as depletion, such as the UF, pH, buffers, etc.
Okay, great, thank you. I think, unfortunately, that's all we have time for today. I know there are a number of unanswered questions, so I'll make sure I liaise with Ramon and Christoph to see what we can do to get back to everybody directly. But I'd like to say a big thank you to both Ramon and Christoph for presenting and answering people's questions today, because we have had a number of other questions from people asking about the presentation slides. I can confirm that if you're interested in a copy of the slide deck, we'll be sharing the email of Stephanie Farrell, who will be able to provide one of those to you, and I've put that in the chat box, but we'll send that to you all afterwards. And similarly, we'll be looking to host a recording of this webinar if you want to go back and review the live materials, and that we anticipate to be available online in around about a week's time. But for now, I'd like to thank you all for your attention and for your questions, and I hope to see you again on another one of our webinars soon.